Hello, everyone, and welcome. I'm pleased to be your host for today's webinar. My name is Dana Jaber, and I'm a Market Development Manager and part of the Global Customer Support team at Bionano. We are delighted to have you with us for the third session of our Bionano in Motion webinar series. This series will spotlight the latest and most impactful studies using optical genome mapping, or OGM, in hematological malignancies, constitutional diseases, and beyond. We are delighted to introduce our distinguished speaker, Dr. Yassmine Akkari, who will present on OGM in constitutional genomics. Dr. Akkari has an impressive resume, so I will highlight only a few of her many achievements. She is a senior director within the Institute for Genomic Medicine at Nationwide Children's Hospital and a professor of pathology at the Ohio State University College of Medicine. She is triple certified by the American Board of Medical Genetics and Genomics in the specialties of clinical cytogenetics, clinical molecular genetics, and PhD medical genetics. Following her postdoctoral research and clinical training at the Oregon Health and Science University, she joined Legacy Health in Portland as the scientific director of cytogenetics and molecular pathology, and recently moved to her current position at Nationwide Children's Hospital. Dr. Akkari has served as the president of the Cancer Genomics Consortium, so the CGC. She also served as the vice chair of the CAP-ACMG Cytogenetics Resource Committee and the chair of the Association of Molecular Pathology, AMP, Training and Education and Program Committees. Currently, she is a member of the WHO Low and Mid-Income Countries Committee, the chair of AMP's Finance Committee, the vice chair of the ACMG International Outreach and Engagement Committee, and she is also a member of the Children's Oncology Group Cytogenetics Committee. Without further ado, please help me in warmly welcoming Dr. Akkari. Yassmine, thank you so much for being with us. The floor is yours.
Thank you so much, Dana, and thank you for the kind introduction. I'm going to go ahead and share my slides, and let's hope that everything works well. Can you see my slide okay? Perfect. All right. Well, thank you again for the kind introduction. It's truly a pleasure to present in this forum and to see all of our colleagues who have joined. Thank you for that. And discuss really some of our experience with optical genome mapping. Although I am presenting a case today in the context, of a single but rather complex clinical case, I'm hoping to convince you of my long-standing belief that every technology in the genomic laboratory is unique and allows for different discoveries, and that one does not really replace another. Between complex clinical presentations and the complexity of the human genome and the alterations thereof, we're finding more and more examples of the value of multiple methodologies in the laboratory. Also, this would be a prime example of the need to be looking at the genome from a structural perspective rather than immediately focusing on sequence variants. This is the case that have surely taught us how complex chromosome level abnormalities can manifest. So as a general reminder, we need to think about the structure of the DNA at different levels. And we have surely learned that any disturbance in the structure of the human genome, whether at the chromosome level, as you see depicted in the slide, or all the way to the nucleotide level, and everything in between, including epigenetic modification, can lead to serious phenotypic consequences and human disease. We also know that there's a plethora of genomic aberrations that can cause disease. And throughout technology development and innovation, we are really trying to figure out the best ways to detect these aberrations and be able to provide an accurate diagnosis in constitutional genomics as well as oncology. So here are some examples of these aberrations. You can see on the left the larger chromosomal aberrations, including deletions, duplications, inversions and insertions, translocations, and isochromosome formation. In addition, there is formation of ring chromosomes, double minute or HSR as a mechanism of amplification. You can see this down in this panel. And all the way really surely to single nucleotide variants, repeat expansion such as Fragile X, and last but not least, disturbance in epigenetic patterns such as uniparental disomy and how those also cause disease. So this slide is to focus on the fact that there is surely a need to develop many methodologies in the clinical genomics laboratories to be able to detect those. So given the wide range of genomic aberrations that exist in human disease, it's important to realize that there has been tremendous advances in our ability to detect them. And more importantly, for our ability to adopt these novel technologies that continue to be developed and will continue to be developed. The only constant in the genomic testing landscape is change. Historically, we were able to see dividing cells. That's how it all started, right? You can see dividing cells under the microscope, and that is in the late 1800s. Understand that chromosomes in these dividing cells carry the units of inheritance. Now, this was followed by a mistake in the lab where we were able to decipher the accurate number of the human chromosomes to 46. Then it progressed our ability to arrange and pair these chromosomes based on size and centromere positions. Then we were able to see more in details the banding patterns through the evolution of banding and staining. Then there was development of techniques such as Baxham-Gilbert and Sanger to sequence at the nucleotide levels. Then in the 90s came FISH for targeting loci and their architecture in terms of rearrangement or enumeration. To then more recently in the chromosomal microarray analysis and the next generation sequencing. And now we are going to talk about optical mapping. All these methodologies are developed to fill a need and a gap in our ability to detect genomic aberrations that cause disease. A very important aspect of this landscape is that we see a parallel technology evolution in both cytogenetics, which is the science of chromosome structure and dynamics, and molecular genetics, which is the science at the DNA level, right, at the nucleotide sequence level. As mentioned in the previous slides, adding hypotonic solution to decipher chromosome numbers, to banding, to FISH, to computer-assisted karyotyping or karyogramming, to CMA, and now optical genome mapping can be compared to the same kind of evolution that we see in molecular genetics from Baxham-Gilbert to Sanger using acrylamide gel. We used to pour these big gels to now automated electrophoresis, to next generation sequencing with massively parallel sequencing abilities, to also development or the acknowledgement in order to analyze these tremendous amount of data from next generation sequencing. We need to develop bioinformatic pipelines to analyze the massive, these massive amount of data. To now we are seeing long read sequencing. It is all really an evolution. And this is really, to me, this is kind of the best lesson that we can gather in clinical genomics laboratories. So why there was such an explosion of genomic technologies? Of course, there is a need, but there's also some kind of parallel forces that took place. Well, we now know that genetic alteration basically touches all aspects of medicine. We see it in neurology, cardiology, certainly oncology, pediatrics, prenatal, and that there are a lot of discoveries that really kind of shone some light onto our profession. And also there's a lot of ensuing media attention that has driven these innovations. So from 2003, when the Human Genome Project sequencing was completed, there was certainly starting to appreciate that all of us have sequence variation, and that these sequence variation also can vary, in terms of race and ethnicity. There's also a huge amount of progress that was done in disease gene identification, such as BRCA1 and 2. We now started to sequence whole genomes in cancer and appreciate what is normal and what is disease driven. And last but certainly not least, the promise of targeted therapy and companion diagnostics. We want to find we want to find what is disease and what is not. We also want to find a targetable lesion, so we can offer our patients drugs that may help them. And so on and so forth. So there's definitely a need to drive these innovations, and the idea is that we need to adopt them. Not only we saw advances in the previous slides in terms of sequencing and targeted therapy, but we also saw an advance in non-invasive prenatal testing, which is now offered not only to high-risk pregnancies, but to the general population, specifically for trisomy 13, 18, 21, sex chromosome abnormalities. And there's certainly recommendation to consider the George syndrome, which is the deletion on 22q. For this test, there are both molecular consideration of the test, itself, but also G-banding and FISH analysis to confirm any of the results that come out of high-risk prenatal cell-free DNA. We know that if there is a high risk for a certain trisomies, most often patients opt to do an amniocentesis and confirm these. Usually the easiest way to confirm it is to do the prenatal FISH or G-banded analysis. And so there's kind of a parallel need in both niches to keep those tools in the genomics laboratory. So let's talk a little bit about G-banding, because this is really what leads to innovation in methodologies. I mean, we have been doing G-banding for decades, and it certainly has served us well. What I wanted to emphasize is that G-banding is not just one test. There's a lot of intricacies, and there are advantages and disadvantages of doing G-banded analysis. And this is what I'm trying to depict in this particular slide. So you can see that our ability to resolve aberrations is very much dependent on the cell type. And the point that I'm making in the slide is that on the top panel, you see this is a triploid product of conception. And you can see that the resolution of the chromosome is about, kind of in the middle range, probably about, 500, 550. On the left panel is a bone marrow chromosome analysis, and you can see that it's very hard to decipher, band by band. Those chromosomes are short. The cells are dividing very quickly, and we really, although we have learned to detect certain aberrations that are specific to the disease that's being interrogated, we can see the challenges of having a huge resolution there. All the way to the panel on the right, which is a blood. A blood, we can probably get to 800 band level, and you can see more material. So the cell type definitely matters. We do need dividing cells, and I'm sure this audience is very savvy about these. We can detect mosaicism because we're looking at a single cell level. Then we can actually understand clonal heterogeneity, which in some cases, in some diseases, is very important. It does give us a whole genome view of a single cell, which is very important. We can see balance, rearrangement, translocation is just one example. However, the last two that I wanted to really discuss is that this is a subjective study, right? It depends on the expertise of the technologist or the team member, right? And so that means that it's very labor intensive. And with the increase of genomic tests, because every aspect of medicine is touched by genomics, we are getting more and more aware that we need to innovate, we need automation, we need less labor intensive. And there's also a big, issue that we're encountering in our field is that we do have a workforce shortage, specifically in the technologist in the genomics laboratories. And so a couple of years ago, we did a study that was born out of a discussion at FACMG that labs were struggling. We were struggling with finding directors. We were struggling with enough fellows that are coming into genomics. We're struggling with the technologist workforce. There just wasn't enough. And so, a study that was led by Marco Long from Nationwide Children's, Sheila Dobbin, and Bob Best, and myself, we sent out a survey to the clinical laboratories and asked them to give us an idea about the current landscape. And I have to tell you, it was a pretty dire situation. And I don't think that we have solved it yet. I think there is an awareness, and that's what we're trying to do. All of us are trying to go out and talk about, educating more in the laboratory sciences, increase interest in genomics and cytogenetics particularly, and so on and so forth. So all this to say really is that we do need a few different methodologies. And so what we want is to be able, and I'm not talking about NGS or the detection of single nucleotide sequence variants. I'm talking about a technology that can detect clinically significant structural variants that are balanced, as well as copy number variants. We need it to be high resolution so that we can see everything in the genome. We need it to be automated, again, because of time and the burden that clinical laboratories have on their staff and their teams. And we also want it to be labor efficient. And so from that perspective, this is really kind of the context of what I want to talk about. So I'm going to illustrate all this by presenting a case example. So we're going to present, in order to illustrate these points. This case, it was actually published recently, and hopefully will shed some light on the many available methodologies in the genomic laboratories, their value, specifically in complex cases, and the lessons that we continue to learn. So the case history, this is a child, a female child born at term, who actually came to attention in the neonatal period due to apnea and desaturation events requiring intubation. She ultimately required actually a tracheostomy in the newborn period and was ventilator dependent at 6 months of age. She had significant hypotonia, but was generally not dysmorphic. She had an EEG that showed no evidence of seizures and an MRI with microhemorrhages in the left cerebellar hemisphere. A sleep study demonstrated severe obstructive sleep apnea. And of note, during family pedigree taking, it was noted that she had the paternal history of polycystic kidney disease. And in fact, when they did the renal ultrasound on our patient, it did reveal kidney cysts. So given the severity of her phenotype, and at that time when this case presented to us, we were not live with rapid whole genome sequencing. It was done in the research environment. We did consent the family, and we did rapid whole genome. And this study on the clinical was done in collaboration between our lab and Dr. Shadori in the clinical and neonatology department nationwide. So from the time that we consented the family to the research whole genome sequencing to the time that we were able to give a prelim was about 4 days. So in this case, rapid genome sequencing actually identified 2 significant events. One is a deletion on the long arm of chromosome 22 that encompasses CHANK3, which is the critical reach for Phelan-McDermid. And this particular slide illustrates the terminal deletion that we see on the long arm of chromosome 22. The second event was a small deletion on chromosome 4, which included PKD2, which explain the family history of polycystic kidney disease. Of note, from our rapid genome sequencing, we did not identify any pathogenic or likely pathogenic sequence variants. So we were really concentrating on these structural variants to explain her phenotype. So let me just pause a little bit about the features of Phelan-McDermid syndrome. So this deletion of chromosome 22 did give the proband a diagnosis of Phelan-McDermid syndrome, which is characterized generally by neonatal hypotonia, which our patient did have, absent to severely delayed speech, developmental delay, and minor dysmorphic facial features as they are listed here in this slide. Most affected individuals have moderate to profound intellectual disability. Other features can include large fleshy hands, dysplastic toenails, and decreased perspiration that results in a tendency to overheat. In general, from a genomics perspective, the diagnosis of Phelan-McDermid syndrome is established by the detection of the heterozygous deletion of chromosome 22q13.3, and you know there's really kind of the paramount involvement of SHANK3. Or sometimes we can identify a pathogenic variant in SHANK3 itself through molecular genetic testing. If an individual's diagnosis of deletion of chromosome 22 was not made through G-band analysis, we usually recommend G-band analysis in order to rule out the presence of a ring chromosome 22 and to see the actual structure of the finding. So when we did the G-banded analysis after the genome sequencing, you can see that this is immediately, you can see that this is not a ring chromosome 22. We did confirm that there is in fact a deletion of chromosome 22 here on the right of the 22 chromosome panel. But what was unexpected in this proband is that there was a translocation, an apparently balanced translocation between chromosomes 3 and 12. And here they are. This is chromosome 3, the abnormal homologue of chromosome 3, if you can see my mouse on the screen, and the abnormal chromosome 12. And so in addition to the deleted 22, this proband had an apparently balanced translocation 312. And so the final karyotype that was reported is that there's a translocation 312 at Q27, Q23, and a deleted 22 Q13.2. And so we were wondering, because of the severity of this child's presentation in terms of the spectrum of Phelan-McDermid, and because of the presence of this translocation, we were wondering if there is more going on in this child. We wanted to look at the structure of the translocation 312. Is it really balanced? Or is there something else that was making the phenotype of this child a little bit more severe than what you would expect in Phelan-McDermid? And so we decided to pursue optical genome mapping. So OGM, just to kind of take a little bit, I know there's probably an audience that is very savvy about OGM, but I'll just, very quickly summarize. It's really the most important, aspect of OGM in thinking about complex cases is that it really, it's an imaging technology, but it provides an unbiased assessment of genome-wide structural variants. So it's not targeted, right? You can see everything. And basically what happens is that you have to isolate high molecular weight DNA. Site-specific labels are applied. That DNA is not digested. It's only labeled. And then the DNA is driven through capillaries, as you see here in this particular, illustration. And the single molecules are detected and the fluorescent labels are measured. There are a lot of quality checks and informatics that are performed behind the scenes. And there are analysis methods that you can use depending whether you're doing a somatic assessment or a constitutional assessment. Ultimately, the maps of the markers are aligned to a reference genome, and it's the discrepancies of these labeling patterns that are identified and can show you whether there's an aberration in that particular genome. So just kind of also another reminder that there are a lot of genomic aberrations that can be detected by optical genome mapping. Using this method, you can identify several types of events outlined here. For example, you can see insertions, and this is, basically the label spacing that increases with or without additional labels detected. It points to inserted sequences, translocations, when one part of the area of that genome aligns with one chromosome and the other point aligns with another. And this is what you can see. You can see here on this upper right panel, and so on and so forth. You can see deletions, which is a decreased spacing of the label, and you can see inversion, compared to the reference. What is also really special about OGM that you can see repeat expansions. And if you have been in a molecular genetics laboratory where you have struggled to first do southern blots for Fragile X and myotonic dystrophy, or if you've done repeat the primer a repeat primer PCR, so you can see the stutter on a gel electrophoresis. Those are pretty tricky tests, and to be able to at least screen for large expansions, using OGM is a pretty significant advance. And in fact, there's certainly a clinical test for FSHD that you could use OGM to diagnose the expansion and the retraction of the area, because it's very repetitive. So I would think that one of the ways to also utilize OGM is in a repeat expansion, at least for a screen. We know that usually males with developmental delay or cognitive impairment are usually screened for Fragile X. This would be a good methodology to use. So going back to our case, what we were able to do is to get blood from not only our proband, because we had already DNA from that, but we also were able to get from the father and the mother. And we recognized that the translocation 312 was actually inherited from the dad, as was the deletion of chromosome 4, which we already knew because there was a history of polycystic kidney disease. The surprise. So I want to just do a little bit of pointing here to the screen. Hopefully you're seeing my mouse. So the mother, we did not identify anything really clinically significant. The father, like I said, the father did have a 312 translocation, and if you can see here, there's that deletion on chromosome 4. The proband had a translocation 312, like we said, the deletion on chromosome 4, but also that deletion on chromosome 22 was actually part of a translocation between chromosome 22 and chromosome 17 on the long arm. And so this is actually a circus plot that enables you to look at the genome in general. So, prior karyotyping of the proband did reveal the 312, did reveal the deletion, but we were not able to, by G-banding, decipher the involvement of chromosome 17 in that particular rearrangement. So this is now showing just the kind of the several results that we were able to obtain from OGM. So we saw the deletion on chromosome 4, and this is depicted here by kind of like a cartoon of the deletion. In red, there's also a CNV analysis that you can see, the visual kind of a user interface that supports a loss on chromosome 4. And then diving into the 312 translocation, you can see, if you look at the middle blue line here, that is the proband's genome, and you can see that the labels continue on chromosome 3 all the way here, and then there is that break point, and then it picks up, and this is the rest of it is on chromosome 12. And so, it's really kind of visually very satisfying to see where these regions map to and be able to really hone down on what are the genes that are potentially disrupted. We really weren't too concerned about the 312 once we found out that that it's actually inherited, but we were very interested in looking at the involvement of chromosome 17 in the Phelan-McDermid. And so this is what I'm showing you here. It's actually the fact that that there was a deletion of the Phelan-McDermid region. And then if you can see here on the top right, this is the actual, there's a gain on the long arm of chromosome 17 that was deciphered by OGM. And then the rest of it is a deletion of chromosome 22. So what is going on with 17? Well, we could not see that deletion. The size was about 800 kb, which is way below the resolution of G banding. But you would think that we would have been able to see it on the rapid genome. Well, we did. We did see it on the rapid genome. It was right here. And on the rapid genome, this is a CNV UI user interface. We can see it, but as you see, when we didn't really interpret it in the rapid genome because it did not include any clinically significant genes. So we kind of said, okay, it's a small gain in the genome. It doesn't have any OMIM or clinically significant gene. We're not really going to worry about it too much, right? Specifically in the context of Phelan-McDermid, right? We never knew until we saw by OGM that there's actually a translocation between 22 and 17 that is causing the deletion as well as the gain in 17q, which really begs the question is that when we see deletions that, at first, sight, they're simple. Unless you do technologies like OGM, you're never really going to see the complexity of these. And this is actually a really cool project to do, to look at these, de novo deletions and see how do they actually form and are they as simple as we think they are. So we wanted, in the technology development laboratories, again, we are able to do long read sequencing, and we wanted to do a little bit of a comparison. The nice thing is that the high molecular weight DNA that you isolate for OGM, you can also use it for long read sequencing, and that is what Dr. Miller did in the lab with, in collaboration with his team, of course. So we used the DNA, and just as an overview of long read sequencing, adapters are ligated to the DNA. In this case, hairpin adapters that you can see here depicted in a slide, where the sequencing primers attach. And this creates a circular template for the polymerase to navigate, and we are able to generate long read sequencing. And so we did achieve about 9x coverage for our patient from long read sequencing. And in fact, long read sequencing did identify the deletion on chromosome 4 for polycystic kidney disease. It did also identify the deletion on chromosome 22. However, from the immediate kind of analysis and the software used, the 312 translocation and the involvement of chromosome 17 was not picked up. We actually had to go to the BAM files and look at the read and see a drop off in the read between, at chromosome 3 to kind of think about the fact that there might be a translocation. So we kind of went and looked for it, right? It wasn't the structural callers that we were using did not identify it. And you can see here we have a drop of coverage, and the reads that align both to chromosome 3 and to chromosome 12 are really allowing us to hone down on the exact breakpoint. And we did the breakpoint actually both from OGM and long read sequencing after we honed down on it, and we're able to confirm the findings. So these are, via this method, we were able really to identify the genes that are involved in all the translocations that we saw. And we can confirm the breakpoints as well. None of the genes, and this is here both from OGM results and also long read results. So the affected gene and also the breakpoint, you can see they're very similar, both for the translocation 312 and the translocation 1722. And this is actually, it's really, the better way to put it is a derivative 17 and derivative 22, because then it's not balanced. You know, the likelihood that the 312 is involved in the phenotype was probably a lot less likely, just because it's inherited. But we were very concerned about what are the genes that are disrupted in the 17 and the 22 translocations. And in fact, when we really looked at the genes, we couldn't find a good answer that, that's why the child is a little bit more severe. But we did find that this deletion is actually a translocation and is more complex. So I wanted to kind of provide now a summary of the detection methods that we were able to utilize, and we're very lucky at Nationwide Children is we do have resources to interrogate all these methodologies and bring them into the clinical space. So in terms of the detection summary, you can see here on the left, this is the cytogenetic locus, so the 4q deletion. There was a loss. These are the breakpoints, and they were in fact seen by the rapid genome sequencing, the optical genome mapping, and the long read sequencing, but it was below the resolution of G-banding. For the 22q, the aberration is actually an unbalanced translocation, really, and we saw it on the rapid genome sequencing. We certainly saw it on OGM, and it confirmed that there is more than one chromosome involved in that. We did when we honed down on it, we can see the breakpoint of it. But in terms of the involvement of 17 and the presence of 312 translocation, really all these, the only methodology that detected all of them was actually optical genome mapping, because these two, we really were not able to see it until we had a priori knowledge of the aberration, so we could hold down on the reads. So the structural callers did not really call it. So I want to give just a brief case summary. So this proband had multiple structural variants identified by a plethora of methodologies, but really optical genome mapping is the one that we were able to truly kind of shine a light as to what's going on. And while her presentation is certainly more severe than what is typically seen in Phelan-McDermott, and the fact that in literature, we combed the literature for anything that is published on that, we were not able to find anything. At the end, Dr. Shadori had a personal communication with Dr. Phelan from Phelan-McDermott, and she did share that she had seen at least 2 such children, and this is thought to be within the expected range of this diagnosis, albeit some children are a little bit more severe. So at this time, I want to switch back to the kind of the philosophy of this particular finding, and really talk a little bit about where do we go from here. I think as I started in the presentation, it's really our ability to adopt novel methodologies to answer questions. We know that the genome is complex. So what are we really looking for that particular project is that we know that several multi-institutional reports have been published on the high concordance between optical genome mapping and the standard of care testing in both prenatal and postnatal study, and as well as oncology. And these are just a few of the PMIDs. So I will stop here and take any questions. Thank you so much for being here.
Thank you so much, Yassmine. We appreciate your time and your expertise in shedding light on how OGM can be used in constitutional diseases. So thank you to all of you for being a part of today's webinar. Have a wonderful day, and we look forward to seeing you in our next session of Bionano in Motion series.
Thank you so much for having me, and thanks everybody for joining. I appreciate it.
Thank you.
Bye.